Hi everyone, I am Devrim Kaya and I am a postdoctoral researcher at the Department of Civil and Environmental Engineering at the University of Maryland, College Park. And my current research is focused on detection of the COVID virus in wastewater influent samples. And in this video, I will walk you through from the concentration step all the way to the RT-qPCR step. So you will learn how to concentrate the viruses, how to convert it to cDNA, and then how to do qPCR so you can quantify the number of the virus particles in wastewater samples. Before I start my concentration, I would like to sterilize all my equipment and my bench so I don't contaminate my sample. Hello everyone. So to concentrate wastewater samples, I'm going to use millipore centrifugal filter units. And the ultrafiltration filter unit, it can filter up to 15 milliliters of the wastewater. It has 100 kilodalton of the molecular weight for the membrane it has in it. And yeah, I'm going to use this to concentrate my wastewaters, which comes, it's like 2 units, and this is the collection part, and inside it has the membrane filters, and you put your water here and then centrifuge this, so your wastewater virus particles or the RNAs and all the genetic material, proteins, going to concentrate in this vessel. So here we go. This is my wastewater sample, and I'm going to first concentrate this and then extract the virus RNA, and afterwards I will do the qPCR, RT-qPCR. So first I want to make sure it is all mixed well, and I will take 30 milliliters for my first step of concentration, which is centrifugation. So this is my surrogate virus. It's an envelope virus similar to COVID-2 virus. I use that virus as a surrogate, and beginning of my extraction or concentration method, I add a known quantity of that virus into my samples, and in the end, during the qPCR, I should be able to quantify it through using primers and probes specific to that virus. So in the end, through the number I quantified in the end, and from the beginning I knew how much I added, so I should be able to calculate how much I recovered. That would give me the efficiency of my concentration and all the way to RT-qPCR method. So now I added the known quantity of my virus, surrogate virus, into my samples, and I'm now gonna go to the centrifugation part to centrifuge my samples. Now my samples are ready for the centrifugation step. In that step I will be separating solids from the liquid part, because the solid would clog the membrane of these filters. So first step is for my methods are centrifugation to separate solids from the liquid, and liquid is going to be concentrated in this centrifugal ultrafiltration unit. Let's go to the centrifugation. So here we go. I have also 2 blanks for each batch of my samples. So I will test if there is any contamination coming from the regions or any steps. So now we are at the centrifugation part, and I will centrifuge my samples at around 3400 G and for 15 minutes. And the important part here, you have to balance the sample, the weight across the rotor. So all my samples have the same amount, so I just have to put them in order, so like that, and this goes here. Oh, okay, now these 2 are balanced and these 2 are balanced. Now you can centrifuge them. Now my samples are centrifuged, and I have the pellet, the solids here, and I'm going to separate the supernatant, the liquid part, into another falcon tube, and that part I'm going to concentrate. So carefully, without disturbing the pellet here, I'm going to transfer the liquid part into relevant falcon tube. Now I separated my liquids and solids. I'm gonna put away my solids into the fridge for later extraction, and I will continue with the ultrafiltration to concentrate my liquid part. I'll continue for the rest of my samples, and then I am gonna go to centrifuge part. Hello everyone, here we are again to centrifuge now the liquid part using this Amicon 15 ultrafiltration centrifugal filters. So the same principle, we have to balance our rotor so our centrifuge doesn't break. I'm going to use the same speed and the time, and you will see the result in a bit. So now we got our samples from the centrifuge, and this part, the lower part, is going to be discarded, and the concentrated part is here. So I'll do the rest and then I'll be back. So now I concentrated my 30 milliliter of the wastewater to 250 microliter. And then I added some water to bring it to 1 milliliter. Now I'm going to wrap around this part, the membrane part, and then I'm going to vortex it a bit, so to get all the proteins or the genetic material stick to the membrane part. So gently cover, appropriately cover all the membrane parts so it doesn't leak, so we don't lose sample. And then I'm going to use the cap, tightly holding the cap. I'm going to vortex it a bit. Side to side. So it should be enough. Now I'm going to use 200 microliter pipette tips with the aerosol barrier because I don't want to contaminate my pipettes. And I'm gently gonna slide my pipette tips over the membrane filter, so I want to get all the genetic material into the liquid part, and I don't want to lose anything. So pipette up down a few times over the membrane. I want to show it to you. So this is the sample. I don't know if you can see. So I just pipette up and down a few times until you get the film over the membrane, which is a bit grayish, darkish. So that is the particles in the wastewater, which has now genetic materials of the virus, COVID virus, as well as other microbes and the viruses. But we are interested in the SARS-CoV-2 virus. I'm carefully sliding my pipette tips over the membrane. Now visually I see there is no film over my membrane, so I'm going to take the extract now into my tube, an appender tube, a sterile tube labeled accordingly. So I'm going to just transfer. So I got everything out, nothing is in it, so I discard my filters. and my sample is 1 milliliter concentrated from 30 milliliter to 1 milliliter. Now I'm going to put into the fridge this, and I will continue with the rest of the samples the same way. So hi everyone, now we concentrated our samples to 1 milliliter, and now I'm going to do RNA extraction. I'm going to use quick RNA mini prep kit, and it comes with a quick protocol, and I'm going to basically follow this protocol. It is a commercial kit. It has columns based, so I'm going to run through my samples through these columns, and then I will centrifuge them, and the RNA will get stick to this column material here, and afterwards I will elute my RNA from these columns. So first I get my samples. I'm going to use 250 microliter. So first I vortex it, and then I get 200 microliter of it, add into my lysis solution. So it has a lysis buffer. I use this 600 microliter of this one. I added into this each tube, and now I am adding my samples. Vortex it, so to homogenize my samples, taking 250 microliter, adding to my lysis buffer, and I will do the rest for my samples. So I added all my samples into lysis. Each sample is in different tubes. I have several replicates here. Now I'm going to mix them like a gently vortex. So at these steps, lysis buffer breaks the cells, and the RNA is in the solution, or the genetic material. All the genetic material is in the solution now. Now, according to protocol, we are going to add ethanol, 100% cold ethanol, and I'm gonna add 1-to-1 ratio to the volume I have in my tube, which is lysis buffer plus sample, and the ratio of the ethanol is gonna be 1-to-1 to lysis buffer. So I'm going to add ethanol to each tube. So we add ethanol now. I'm gonna mix it. Now we are at the column step. I'm going to transfer all the mixture from this tube into the Zymo column collection tube, and then I will centrifuge it. I'm going to do the same thing, transfer all the samples in 2 rounds into my columns, and first I centrifuge, and then discard the liquid here, and then add the rest of the lysis plus samples plus ethanol into the column. Now I transferred half of my samples into my columns, and the step to centrifuge it. So centrifuge is done, and now I'm going to remove my samples. As you see, it has a liquid part. Now it's filtered, and I'm going to discard the flow through, and I'm going to keep the column and load the rest of my sample to the column, and I'm going to centrifuge, do the same thing. Yes, as I said, our sample is centrifuged through the columns. Now I'm going to discard the flow through and keep the column for each of them, because I'm going to load the rest of my sample to here, and this column is keeping my RNA holding my RNA. RNA is attached to this column. Loading the rest of my sample. I will do the same thing for the rest of the samples, and then centrifuge them again and discard the flow through, and then I will move to the washing steps. So now I successfully centrifuged all my samples through the columns and discarding all the flow through, and my RNA is in the column, and I'm gonna move the next step, which is in that step we are going to use the Rna Prep Buffer. Now according to this step we are going to add 400 microliter of Rna Prep Buffer to each of these columns. So I need to move to Prep Buffer. Okay, another Prep Buffer. It wasn't enough, so I'm going to get 400 microliter from this one. Now I'm going to spin down them again for the next step. I spin down again, centrifuge them. Now the next step is washing the RNA, and it's a reagent with ethanol. It's a pre-prepared by Zymo, so it's going to clean up the RNA in the column, and it's going to make it ready so we can elute it with the RNase free water. It's removing all the inhibitors, salts, humic acids, so it's cleaning up our RNA. So whenever I feel like my gloves are not clean enough, I want to clean them with ethanol. So next step is 700 microliter of Rna Wash Buffer. Going to add 700 microliter into each column. Ready for another centrifuge at the same speed and temperature. Ready for the last step of the wash for the column, and this time I'm gonna add 400 microliter to the same columns we washed before, and afterwards we are ready for getting our RNA out of the column. Now we are going to centrifuge our loaded columns, and we are going to discard the flow through collected into these collection tubes. So the important thing here is putting the tubes evenly across the rotor, so the weight is distributed evenly and balanced, so we don't disturb the rotor, break the rotor. It's important. I don't know if you can see it, but sorry for the technical difficulties here. Loaded, closing, and the speed, the time, and the temperature is 4 degrees because we don't want to destroy the RNA. This time I'm gonna spin it 2 minutes and ready. Centrifuge is done. Now I'm going to open and take the samples out. So you see it's filtered. I'm going to discard the flow through and keep the column. So this is the last step. We centrifuge it, and we are going to now transfer our columns into clean microfuge tubes, because I am gonna elute RNA from the column into this collection tube. And this one goes to trash. You don't need flow through. So following our protocol, now we are at the elution step, which is the last step here, 8. So to elute my RNA from the column, I'm going to add 100 microliter of DNase, RNase free water directly onto the column matrix. The column matrix you see here is the white part of the column. I'm gonna add my RNase, DNase free water directly on top of it, and then I'm going to centrifuge it again. And this kit also comes with RNase, DNase free water. I am going to add 100 microliter of this water onto each column matrix. I'm going to use 100 microliter of the pipette filters with the aerosol barrier, because we don't want to cross-contaminate our samples and our water. So yes, now we are going to elute our RNA from the column matrix. For this I'm going to use DNase, RNase free water, 100 microliter directly over the column matrix. If you want more concentrated RNA, you could reduce the amount of RNase free water to 50 microliter, so you get more concentrated RNA. So I'm going to do the same thing to each column, add RNase, DNase free water onto each column, and then I'm going to centrifuge it. Now our columns are ready to be eluted, so another centrifuge step. Centrifuge is completed. As you can see, we eluted our RNA. Now RNA is in this liquid. So yes, now we eluted our RNA, and I'm going to discard the columns now. I don't need them anymore because I got my RNA here. Okay, we got RNA here in these tubes. Now I have to keep them on ice because RNA can degrade very fast. So I have my ice here. I'm going to put them there, and I'm going to measure their concentration. So I need a blank, which is going to be our matrix, which is RNase, DNase free water, to measure the concentration. So I'm going to use nanodrop here. So now we got our RNA eluted and on ice, and now I want to measure its concentration. I'm going to use nanodrop 2000, and it is a thermoscientific brand. And for this I open the nucleic acid, check the arms are closed, and now I have to blank before measuring my samples. Yeah, so I got my 2 microliter of matrix DNase, RNase free water. So I loaded the 2 microliter of the water here and closing down arm gently. And now I'm gonna blank. Now we can load samples. So I'm going to label them here. going to measure sample 1. So same thing, I'm gonna use 2 microliter and measure. Okay, let's do this way. So I got my sample and got my 2 microliter pipette and tip. I'm gonna get 2 microliter and going to measure it. I load my sample. So now we loaded our samples onto the arm, closed the arm. Now we can measure the samples. When I labeled my samples, now when I press the start, it's gonna ask me to give a file name and to save the file with all the data it's gonna measure. So let's label it, give a name to the file as well. It's gonna give me concentration of the RNA and ratios. It isn't great, but it is okay, it's gonna work. So I finished measuring all of my samples, and now I finished measuring all my samples, and I would like to export my data. So I go to reports, and from there I choose export in XML, and I give a name to the file. Then I can reach it whenever I want to. So now we extracted our RNA and measured them. The next step is to convert the RNA to complementary DNA, which is cDNA, so we can quantify it through the RT-qPCR. So we are going to use reverse transcriptase enzyme and follow a protocol to convert our RNA to cDNA. Our protocol I'm going to follow. In the first step, I'm going to make a reaction mixture. I'm going to call it master mix 1 here, you see, and I will use random hexamers, DNTP mixtures and water, and then I add 6 microliter into each tube, and then I will add another 6 microliter of the RNA, so total volume will be 12 microliter in each tube. And then I mix it, and then I will do at 70 Celsius degree denature my RNA for 5 minutes, and then I'm gonna cool it down. So in the second step, after cool down, I am going to make another reaction mixture. I'm gonna call it M2. In this one we have buffer. This is the reverse transcriptase buffer, and these are the volume for 25 microliter. My total volume in the end is gonna be 25 microliter, so I calculated for 25 microliter. And superase is an RNasing enzyme, so it would inhibit the RNase, which if we didn't use it, it would degrade our RNA. And then I'm going to add DDTs. This is our reverse transcriptase enzyme, and water, bring it to the total 13, and I'm gonna distribute into my tubes which had my mRNA and my reaction mixture 1. Then the next step would be incubating the samples at 42 Celsius degree for an hour. Then the following step is inactivating enzymes at 70 degree for 20 minutes, and then I will have my cDNA, so I can keep it at 4 degree. So in this hood, all the materials and the tools and the pipettes and these PCR tubes, after I clean them with the ethanol, sterilize them with ethanol, as well as using RNase away reagent. So I try to sterilize and remove any inhibitors on my equipment tools. Now it is being UV sterilized, and UV would degrade if there is any DNA or RNA. So hopefully I will not get any contamination in my regions. So here we have our RNAs extracted, and the regions are melting on ice, which are needed for cDNA synthesis. So we are gonna convert our RNA to cDNA using all these regions: the NTP, random hexamers, and the buffer, reverse transcriptase enzyme, and the superase is RNase. So this is an enzyme called superase, which is an RNase inhibitor. RNase is an enzyme would degrade RNA, so we don't want that. That's why we are gonna add that RNase inhibitor to our mixture. So I prepared my master meat, first one, and distributed 6 microliter of it into each tube, and I added 6 microliter of the RNA into each tube, centrifuged it. Now I'm going to do, using the thermocycler, going to the first step of the cDNA synthesis. Now the run has started. Now I'm gonna go to make my master make 2. And for that one, my regions are here: buffer, superase enzyme, DDT, transcriptase, and water. And these are the volumes I'm going to add. And all the regions and the RNAs are here. I'm done with the RNA. In the second stage, here I'm going to make a master mix and then add 13 microliter into those tubes. Now first step of the cDNA completed. I'm gonna remove my tubes, keep them on ice. Now I'm going to add, I already prepared my master mix 2 according to this recipe. Now I'm going to add 13 microliter of the master mix into each tubes, and I'm going to add 42 Celsius degree for an hour, and then it will follow this step, inactivate the enzyme at 70 Celsius degree for 20 minutes, and then it's going to keep it at 4 degree until I remove them from the PCR machine. Let's continue. Now I added the rest of the reagent, master mix 2, including the reverse transcriptase. I want to quickly spin it down. Going to choose cDNA second step, loading my tubes, making sure the lids are closed. And yeah, this is the temperature method. I'm gonna hit run. So now it shows it's gonna take 1 hour 20 minutes. So the last step is 4 degree. I don't have to wait here, so I'll come back, and when the method is finished, I'll take my samples out and put into the fridge.